Thanks so much for inviting me, Mission Bio, and thanks so much for coming, everyone, and sticking around so late. So I tried to make this as entertaining as I can, this being 8:30. I want to really put some attention on the fact that our blood system needs to generate hundreds of billions of cells every day. But ultimately, all of those cells derive from just 100,000 stem cells, which is really not a lot, not a very big number. So what we're trying to do is understand these 100,000 stem cells as individuals. And one thing that the field knows is that during aging, some of these 100,000 stem cell clones that we have when we're young expand and start to dominate blood production, whereas others appear to, well, disappear. And these clonal expansions go along with biases towards making more pro-inflammatory immune cells at the expense of B and T cells. So this weakens immunity. And also these clonal expansions are, of course, the first step in leukemia formation. So how can we track and characterize these HSC clones without genetic mutations? Now, the first thing that would probably spring to the mind of most of you would be clonal hematopoiesis mutations. But one thing that's really important to recognize, and I will be showing data on that later in my talk, is that maybe the red clone has a clonal hematopoiesis mutation, but the blue clone has not. And there's actually epidemiological data that shows that the health consequences that you associate with clonal hematopoiesis, they are actually effects of clonal expansions and not of the driver mutations. So I really want to challenge this concept of looking at driver mutations and rather focus on the clonal expansions. But how can we look at these clonal expansions if those cells have all the same genome? Of course, they have point mutations, one place or the other, but we don't know where, right? So we need something that mutates super rapidly and that we can read out very, very easily at a high scale. And maybe those of you who are deep in the field know about people working with mitochondrial mutations. Now here we're presenting something different, which is epimutations. So as our DNA gets copied, so does the DNA methylation pattern. This DNA methylation pattern gets established sometime during embryonic development, and then it's like, in some regions, of course, massively regulated, but in other regions of the genome, it's just passively maintained. So as the cell divides, the DNA methylation pattern gets copied over by an enzyme called DNMT1. And this is an error-prone process. So you sometimes lose methylation, and I think sometimes you also gain methylation. To use epimutations for identifying clones, you need a way to read out the methylation state of single CpGs at the level of single cells, right? So most single cell methylation methods don't do that. They offer super sparse coverage. They are technically difficult to do, but they also offer super sparse coverage. So there's no way to really look at the effect of a single CpG, but this is a stochastic process happening at single CpGs. So what we needed was a method that does that, reads out the methylation status of individual CpGs. And we came up, well, back then we didn't think about epimutations, I have to say, but working with a group of René Beekman, who has an interest in methylation biology, we came up with this idea of doing a targeted analysis of methylation on the Mission Bio platform, which how this works, where you all know the Mission Bio workflow very well, right? Like around up to 1,000, at least a few hundred, DNA targets get amplified by a PCR and droplets. And the only modification we did to this workflow was that we added, prior to the amplification, the digestion with a methylation-sensitive restriction nuclease. So what this does is that it cuts in a certain context if there's no methylation, but it leaves methylated DNA intact. So basically, this means that the unmethylated DNA does not get amplified, whereas the methylated DNA gets amplified. And you thereby get, and this is really the important message, you get a binary matrix of DNA methylation, where with one experiment, you cover around 10,000 cells, right, like Mission Bio always does. And on the other dimension here, the rows, you cover up to 1,000 CpGs. Now, before I go into more detail on the bio, first thing I should say, this is very easy to use because it's literally just add an enzyme to the workflow that you all know. it has a very, very low noise. You all know that for genotyping Mission Bio, I mean, it covers the NMP3A site that you're interested in really in like most of the cells, right? And the same happens here for methylation. We quantified this in a lot of detail. I don't think I need to walk you through this, but basically we have a false negative rate of 7% and almost no false positive rate. If you contrast this with any other genome-wide single cell methylation method, it's like an order of magnitude essentially difference. Same as out of the box Mission Bio, this method can be combined with surface protein expression profiling. Here on the left, I'm showing a UMAP that's actually done on the methylation. Give a bit more insights about this later. So you here see this is mouse hematopoiesis. You see hematopoietic stem cells differentiating into different lineages. And on the right, I'm showing the antibodies, the surface antibodies that the people in mouse use to define stem cells, in particular CD48 and CD150. And from the colors, you can see like that these populations really match extremely well, almost as if the thing on the right were FACS data. So there's protein expression. Of course, we can do the thing that Mission Bio is really good at doing to begin with, which is profiling clonal hematopoiesis mutations in the same cells. And things that I might show a little bit, but Lindy already talked about it. There is a possibility to profile RNA together with this and for the aficionados mitochondrial genomes. But now let's really get into the biology. What did we do with this? So we had this method, and we were interested in applying this in the context of clones, hematopoietic clones. And for this, we first of all needed a ground truth. So we started in mouse, because in mouse, what one can do is one takes a population of HSCs and with a lentivirus introduces a heritable DNA level barcode, does a transplantation, harvests cells 12 weeks later, and then we perform this assay and we read out this Larry barcode also on the DNA with Mission Bio as if it were a mutation, right? And when I first looked at those UMAPs, I knew that this was going to be a big story because this is nothing like what an RNA-seq UMAP would look like. So on the right, I'm showing differentiation. This is essentially the colors come from the surface antibodies. So we know this is myeloid precursors, red blood cell precursors, stem cells in between. And clearly the methylome captures this differentiation state, but it captures something else. I always like to say that these UMAPs look like spaghetti. And if you pay attention, you will notice that this spaghetti here has one color and they all have the same Larry barcode, which means that these are all the daughters of one stem cell, right? So this one stem cell gave rise to red blood cell and white blood cell precursors, but the methylome captures that those cells are cousins or siblings, which is something that on any other modality that I've ever looked at is not, this information is just not retained, right? This is actual lineage tracing. So then we asked whether these are maybe different parts of the genome that capture differentiation versus lineage information. And this actually is the case. So we just check for each CpG if it's associated with any surface protein. That's a sign that it's differentiation associated. So if we take these CpGs that are differentiation associated, we make a UMAP, it looks like RNA-seq. It gives you differentiation. Okay. More interesting, if we do a UMAP on the CpGs that are not differentiation associated, obviously it does not capture differentiation, but it captures clone. So this basically, the colors here are now the Larry barcodes, the ground truth clonal labels. In color shown any clone that's bigger than 0.25% of the blood system, and they are captured with over 90, well, almost in our quantification, close to 90% accuracy, but the Larry also makes mistakes. So I think we're getting this extremely well. So this was, we then did some applications on mouse aging, which I won't talk about, because what my team in particular was really excited about was, I should have said the mouse experiments, I will say it later, but the mouse experiments were a collaboration with my colleague Alejo Rodriguez-Fraticelli at the IRB. Like, I'm not a mouse lineage tracing person at all, and without him, we obviously couldn't have done this at all. But my team then was really eager to translate this to human. So the first thing we needed was a new methylome panel. I deliberately didn't say how we did it in mouse. You need to define some CpGs, right? It is a targeted method. But what we learned along this project is that we don't actually need to worry too much. So basically, we need some CpGs that are variable. We don't want the ones that are always methylated or the ones that are always unmethylated, since those don't have any information. But we did this a huge exercise in mouse to define really informative CPGs. We made it an even bigger exercise in human, looking at CPGs that are differentially methylated between stem cells and progenitors that are variable across individuals or within stem cell populations, all from bulk data. But in the end, the only thing that really matters is that the CPG is like not fully methylated or not fully unmethylated. Any other CPG has information, or at least that's what we see. So this is part of our panel. We have this quite optimized now. We now have a 300 target panel that we work with. And that we know that all of those CPGs have good information, and that they work technically. So then we added, in the human context, a genotyping panel for these clonal hematopoiesis mutations. This is essentially a subset of the standard mission panel. Okay, so we applied this to bone marrow from aging individuals, and the first thing was, of course, to capture cell state. You've seen this from mouse. I'm not showing this again in human. This is total bone marrow samples. Some data integration between antibodies and dynamic CPGs doesn't matter. Point is, it resolves cell states pretty well. Now comes the exciting part. Now we looked at these static CPGs, and this very busy slide. We have 7 individuals here, all sampled total bone marrow. The youngest individual is 22. The second youngest individual is like early 50s, and then we go up all the way to 77. And don't focus on this one, we didn't sample enough cells. But if you look at the other individuals, what you notice is that the older individuals get, the more clonal clusters get. Right, so it's very clear. For example, here, this individual, age 75, if I'm remembering correctly, there's several clear clusters of clonally related cells. And this big cluster here additionally carries a loss of Weyermann. So this gives us like it's sort of a ground truth, right? Like there is a mutation in one of the clusters, but this is what I was saying earlier. Here we have, for example, the clonal hematopoiesis mutation, ASXL1, yes, we see this as a clone, but we see many other clones that don't have any mutations. And some of those samples were extensively pre-screened for clonal hematopoiesis mutation by Parrish Vyers and colleagues. So we know exactly what is there. And we know that we're not like, surely we're missing mutations, but we're not missing any of the mutations in your favorite clonal hematopoiesis genes. So with age, we quantified the sensitivity of this assay as 1%. So we detect in human clones that are bigger than 1% of total. And those clones that basically at an age of 50 or so, this seems to start, and then it seems to linearly go up in age. And the older you get, the more expanded blood stem cell clones you have. So that's great because it allows us to quantify clones. Of course, one could, well, you can grow single-cell derived colonies and do whole genome sequencing if you have the money for it. You can sort of make guesses about this from whole genome sequencing, deep whole genome sequencing of a bulk sample. But I think this is a really powerful method for even just describing that process. But we can do more because we have the whole, well, antibody profile, CPG profile, so we can look at what these clones actually do. And to that end, I basically grouped the cells in bone marrow into 2 groups, the ones that belong to expanded clones. So we have evidence that they are from a clone that's bigger than 1%, and the non-expanded clones. And then, of course, for each clone, we could also quantify whether it makes predominantly red blood cells, white blood cells, B cells, T cells, etc. And what we saw, let me show the data as well. If we compare expanded versus non-expanded, the expanded ones go up, and well, they go down in erythroid production and down in B cell production, which means they go up in myeloid production. And then now comes the twist. Within the expanded clones, there are the ones that have clonal hematopoiesis mutations and the ones that don't. We can compare those. And what we will see is that the clonal hematopoiesis mutations, they make this trend even stronger, but they are not a game changer. So yes, the CH mutant clone is a little bit bigger than the other expanded clone. It's a little bit more myeloid biased. At this CD34 stem cell level, we somehow see this as significant HSC enrichment, what we don't see for the other expanded clones, but I think that's detail. The take-home message, or at least what I take from this, really is, let me skip this, come right to it. So these CH mutations might not really be the primary cause of anything here. They might rather be a symptom of some aging process. that happens, maybe driven by the microenvironment, by inflammation, we don't know. But there's this epidemiological data that I mentioned earlier that it's clonal hematopoiesis, it's clonal expansions and not CH mutations that matter for these health outcomes, like heart disease risk, like leukemia risk, like overall mortality. And I think with the method that we have developed, this epiclone, is really the first time that one can look at the stem cells from these expanded clones without having to rely on the mutations. So I think that's mostly what I wanted to say. The study is published since a few weeks ago. I think I'd skip over I just show one more. Where's the mouse? Here. Just show one more slide that we have two additional technical capabilities, which I already mentioned, which is that we can add RNA to this assay. The implementation that we have is probably not as easy to use as the ones that you guys developed. I loved seeing that you add the RT to the protease mix. I think this makes things much easier than what we did. But it's obviously it's nice for characterizing these clones and then really important moving forward. And as I said, for the people who are deep in the lymph tracing field, we can also genotype mitochondria. So with this, big thanks first of all to the team who did this project developed very fast after we had this initial discovery. So at some stage was a really large part of my life working on this full time. The people shown above, especially Michael Scherer, the postdoc who led it, Martina Braun, PhD student who then did all the human analysis. Alejo and his PhD student Indra, without whom we would never have started this, and who did all the mouse work. And especially during the revision process, Mission Bio was a fantastic support because we had to do a lot of Mission Bio runs after we had support to submit this. So thanks a lot for listening and happy to take your questions.